I'm Jonathan Carter of the Autism Science Foundation, and welcome to the ASF Science and Sandwiches event at the Seaver Autism Center at the Mount Sinai School of Medicine. First of all, I'd like to thank the entire Seaver team, especially Jessica Brownfeld, for their great help setting up the event. Science and Sandwiches was formed to bridge the gap between scientists and the individuals and families affected by autism, and it was established, of course, to eat sandwiches. With that said, I can think of no better researcher to present at this event than Dr. Joe Buxbaum. Dr. Buxbaum, the director of the Seaver Autism Center, has been at the center for over 15 years, and as a world-renowned molecular geneticist, he hopes to understand the autism spectrum on the molecular and genetic level. His research one day may lead to the development of treatments for ASD. Dr. Buxbaum has laboratory molecular neuropsychiatry and uses animal models in order to evaluate treatment approaches. He has created mouse and rat models of one of his most promising focuses, the SHANK3 gene. SHANK3 affects individuals with Phelan-McDermid syndrome, a type of ASD. And without further ado, Dr. Joseph Buxbaum.
Thank you, Jonathan. So I'm going to kind of survey some of the work that I'm most involved in. And the idea will be to kind of show you how our view is that the basic science will actually lead to changes in treatment that will benefit families directly. The Seaver Autism Center is a fully integrated center. It does a lot of things, and we all work together kind of with a single mission. So it's quite a tight group of people. And as I've said more recently, that when you get the psychologists and the clinicians and the clinical researchers and the imagers and the basic scientists all together, and you make them talk to each other every day for about 2 years, then about that point they begin to understand each other enough to think kind of collectively about moving this issue forward. So these are some of the things we do at the Seaver Autism Center. A lot of research, but clinical research, clinical trials with drugs, clinical trials with behavior, imaging, model systems, molecular research. I'll talk a lot about these three: social skills, medical management, IEPs, detailed assessment, and genetic counseling. Those are just kind of to give you the broad flavor, but I will not talk about all of that. I'll talk about kind of the more molecular and genetic parts and how it can lead to better model systems and then ultimately clinical trials. So, and the points I often make, and I will make today as well, is that, and this is almost a minor point, but it's good to bear in mind when I get further into gene discovery, that autism spectrum disorders have significant heritability. A lot of the risk is accounted for in genes. And the key thing is identifying a cause of autism, be it a gene or something else, will have profound impact on the family, short and long term, in my opinion. There has been an enormous amount of gene discovery in autism. We think we probably have about 15 to 20% of the genes identified, and we know exactly what we need to do to find the next number of genes. But we have very good estimates of power. And I will talk about this only kind of by inference, but as we find these genes, they implicate high-value pathways for drug development. So this is just because I should say it, but it's also, I don't need to explain to you all what autism is. It has these deficits or alterations in three domains: social, language, and the presence of repetitive behaviors or stereotype patterns of behavior or interests. The key thing I want to make here is that it's a behavioral diagnosis. So when somebody has an autism spectrum disorder or condition, it says nothing about the cause of the disorder. It just says this is a manifestation of what you see in front of you. And the reality is, and the evidence is absolutely unequivocal, that the autism spectrum conditions are manifestations of multiple independent sorts of genetic or molecular conditions. So I say before and after here, but really what I mean is if you look at it from the point of view of the behavioral phenotype, you might say that all the kids on the spectrum share certain properties. If you look at it from the molecular perspective, you may see very different things. You may see that some kids with autism have certain changes, some kids with autism have another kind of change, right? And it may be that drugs that work for the kids that have, let's call it the yellow autism, may be much less effective for kids with orange autism and vice versa. So this is this idea that's emerging of called personalized or individualized medicine. The idea that you want to choose a drug to target a specific cause of a disorder. Now obviously, EI and behavioral interventions, that will work equally well to the best of our knowledge for everybody. But if you think about pharmacology, it may turn out to be that you really need to target drugs to the patient itself. And this is kind of another way of saying exactly the same thing. This is from an old review, but basically it has a series of genetic conditions associated with autism, and then it asks two questions. Question number one is, if you had 100 kids with autism walking in your door, what proportion would have that syndrome? And you can see somewhere between rare and 1% to 2% for the typical medical genetic condition with autism. People know that Fragile X, for example, is associated with autism. We work on this gene here, which is strongly associated with autism. The other way to look at it, and this is what I would call the bottom-up way, is to ask how many kids with a SHANK3 deletion have autism, right? So here you're collecting your sample, if you will, or your study group from the point of view of the genetic cause, while here you're collecting kids based on phenotype and asking what proportion have a genetic cause. Does that make sense? So you can actually look at the disease from two different ways, right? Or the conditions. And so I'm going to take, so if we just kind of take it more or less on faith that these are a small number of probably about nearly 100 different genetic causes that contribute to autism risk, you know, the question is what can we do with that information? And the question I'll talk about very briefly at the end is how do we get to the next level in terms of identifying more genes? These multiple causes of autism, to find them, I use genetic methodology. And the reason is that there have been now, this is 8 studies, but there's now a 9th study that show that autism is quite heritable. So I don't think anybody really challenges the notion that autism is highly heritable. There was a paper that came out last year by Hallmeyer et al. That said it was a little bit less heritable than we thought, which is not the same as saying it's not heritable. Many people, for a variety of good kind of statistical and epidemiological reasons, are concerned about that data set. But it doesn't really matter for me. Blood cholesterol, for example, is much less heritable than autism. It's about 30% heritable, while autism, in my view, is probably 80 to 90%. It doesn't mean that you can't find a good drug for cholesterol using genetics. Even if something is heritability is as low as 10 to 20%, as long as it's not non-zero, the methods of genetics will find you genes. Drugs interact with genes. They don't interact with, as much as I value environmental research, and we do it extensively, for example, in other disorders I work on, you can control the environment, but you can't make a drug for the environment, right? So if the environment is a major part of a certain phenotype, like lung cancer, you can control your smoking, right? If it's a risk factor for bladder cancer, which it is, you can control some of the foods you eat, right? But when you think about a drug interacts with genes, and so finding genes that underlie a disorder actually turns out to be a good way to identify novel drug targets. So this is really, and as I said, there's one more paper. Even the Hallmeyer paper is on here, it's just cut off. The key thing about this is that whenever you have somebody who's genetically identical, a monozygotic twin, they're much more likely to share the autistic phenotype than if they are siblings or dizygotic twins, right? In every study ever done, this number over here for MZ twins is bigger than that number, including Hallmeyer. So nobody doubts that genetics is a fairly significant thing. Whether it's half the risk or 90% of the risk is really, you know, in biology, we're happy if we just get the order of magnitude, as we say, correct. Getting at something within twofold, boy, that's unusually good. All right, so we think there are genes to be found. We know there are genes to be found, and we can invest our resources, your resources, in finding those genes. And the question is, why bother, right? And I think there are 4 kind of reasons. I mean, people talk about 3 things: benefit to the child, benefit to the family, benefit to society. Those are the 3 sorts of benefits that go into the concept of genetic testing altogether. I didn't break it down quite that way, but let me just go through these 4 and you'll see how they kind of map. So first of all, the family learns something specific. They understand about etiology. They understand how this happened to some level. You never fully understand, right? But at least you understand that it wasn't, you know, my family had some severe illness, and, you know, for reasons that escape me, People in my family thought it's because my brother fell and hit his head when he was a kid. That wasn't the reason, but it was just kind of the family narrative, right? And it didn't, I couldn't change it, right? It was just kind of, having an understanding, we are people who like to understand our world. So, but the other part of this understanding is recurrence, which means that we all have heard that the risk for having a second child with autism is 8% by the old studies, with the baby SIBs as much as 18%, right? Genetics can actually tell you something more specific. So if you have a child that has a certain kind of new mutation that wasn't found in mom or dad, that family doesn't have any increased risk for autism, right? So you can learn something about recurrence, which is really kind of what most people think about for genetic counseling. Advocacy groups are a very important thing. So you've heard about the clinical trials in Fragile X syndrome. Those are driven largely by families who got together because they shared a diagnosis, a molecular diagnosis. They pushed around developing drugs for that, let's call it subtype of autism, and then they actually funded it, right? They raised the money to get the first Fragile X trials. In addition, these families get together and they say, hey, I'm taking this. I found that this is very helpful for my kid, right? So a lot of the kind of the peer-to-peer interactions around, what to expect, what might work, what doesn't seem to work, may in fact kind of, is always shared amongst families. But if there's a shared molecular diagnosis, that sharing might be very important to understand things, a better sense of the process, right? Model systems, which basically means if you have once you find a gene, you can mutate that gene somewhere, either in cells in petri dishes in the laboratory or in mice, and then you can understand something more about the pathophysiology. And I'll talk quite a bit about that. And then, as I said, every gene is a therapeutic target, right? It may not be a good one, but if you find 10 genes, within them there are probably a couple that are what we call druggable, suitable to be treated with drugs if you develop the right drug. So this is why when you find a gene, you have all these kind of potential benefits to the family, but to the society on the greater level. And this is kind of an example of one of those things I just mentioned, which is now in routine use in many sites. This is called array CGH, and it looks very quickly and carefully at the chromosomes of an individual and asks questions about changes in the chromosomes. So this boy has a little tiny deletion on chromosome 15. It's kind of blown up over here. And as I said, that gives, first of all, etiology. It tells the family about something causal. This was a de novo mutation, so it wasn't found in mom or dad. It was found during the creation of a specific egg or sperm cell. So the family has no additional risk for autism because it's kind of a one-off. And it contributed to the literature and our understanding because this area has actually been implicated in autism before, but this boy, by the nature of the deletion he has, actually showed us a very small number of genes which were likely to be the main genes involved in this kind of autism. So it added to our knowledge base of what are the genes that contribute to risk. So, and I'm going back, so that's kind of about recurrence and about kind of clinical information. But the other thing I want to talk about is this idea of model systems and therapeutic targets. So this is this concept that's become very popular now, which says that if you find a gene or a causal component to a disorder, then you can model it. So here are some mice, but you can do it in rats or you can do it in cells in culture. And basically the idea is we now have the technology. With mice we've had it for almost 20 years, but now we have it for rats too, where if you can take any gene in the mouse and mutate it. So if you find, for example, the FMR1, the Fragile X gene, and you want to understand more about Fragile X, you mutate that gene in a mouse, and then you begin to study the mouse, and the mouse will begin to show changes, and this is the connection between nerve cells and the brain. You'll see changes in that mouse with this connection of nerve cells in the brain, which will then kind of lead you to think about drugs that may reverse that deficit, novel drugs that can then be tried first in the mouse and then in patients. And that's really what happened in Fragile X syndrome. So they created a mouse, they found the gene FMR1, they knocked out the gene in the mouse, and they discovered that certain processes in the brain were slightly different. And we know enough about brain function to be able to have said in that case, well, these drugs that are actually already have been made by pharmaceutical companies may actually be beneficial, and they were tried in the mice, and sure enough they work, and that's how we have some of the clinical trials that are going on right now. Right? So that idea that finding a gene leads directly to a mouse with the same mutation and then ultimately, hopefully, to novel drugs is very kind of widespread. And we all actually think that in the case of neurodevelopmental disorders like autism, we are going to lead the pack. We can talk about why later, but we have a lot of leads. So this is 2 quick slides going back to that original slide I showed you, and everything that's underlined in red, somebody has made a mouse with that mutation. There are more. There are literally hundreds of mouse mutations that correlate with autism genes, but just from that one slide, you can see that almost every gene, and this one in yellow we did, and we did one over here too, but almost every gene or locus has been mutated in a mouse. And you can learn an enormous amount from looking at those mice, besides the fact that they're very cute. So those are some of the mouse models. And the next question is, how many of those have actually translated into at least a first pass clinical trial? And the answer is, I don't know, but I know of these 4. TS, Rett syndrome, Fragile X, and I'll talk about this because this is about to get started. So every mouse here, almost without exception, has been made. And amongst those mice, some of them have led us to a first-generation clinical trial. All right, so I'm going to talk a lot about this, probably way too much about this, and give you specific examples from our laboratory. But I've kind of given you the big picture right now, and now I'm going to drill down to some kind of painful nitty-gritty. So questions now before we? Yeah, go ahead.
When you say you're making a drug, is that to cure, or is it to just cover up or deal with symptoms? What does that mean, a drug?
Well, so, as you know very, very well, the drugs for autism today are dealing with what you might call things that are not the core symptoms of autism. We're not here to make a Me Too drug that copies some of the drugs that are out there. That's not what academic science does. We're trying to bring the next generation of drugs that actually deals with core symptoms of autism. At Mount Sinai, we think about behavioral interventions and we assess them because those actually address core symptoms. We're also working with oxytocin on another compound that we think will address core symptoms of social interaction. We have a lot of data on oxytocin. And here, what we're looking for the next generation of drugs are things that actually are disease modifiers and actually address core symptoms. I'm not going to compete with pharma about making yet another kind of drug that's kind of like the other one. Yeah, they do that really, really well. All right. Yes.
Yeah. When you speak about de novo genes.
De novo mutations.
I'm sorry, de novo mutations. Are there circumstances that can create these de novo?
So we all have de novo mutations in our bodies, right? So it turns out that we've just finished a large study on looking for point mutations in people, and everybody in this room has at least one coding mutation in, and probably 8 to 10 fairly large de novo CNVs, and on and on and on. De novo mutation turns out to be part of the human condition. It's part of our diversity. If it hits the wrong gene, then you start to get consequences. We don't have any evidence yet of anything that causes excess de novo mutation. There are some genetic findings that suggest that certain loci in the genome may be more prone to de novo mutation depending on other genetic loci, but we don't actually have any there's no evidence for example, an environmental cause that increases de novo mutations besides, huge amounts of mutagen. All right? Does that answer the question, more or less? We can come back to it. All right, so I'm going to talk about this, as I said.
One more question.
Yes?
What sort of behavioral changes do you see in the mice where you've replicated the...
Why don't I show you with a specific example? But let me also preface that by saying I'm always pretty underwhelmed with mouse behavior. I'm much more interested when I think about, the Fragile X drugs were not made because the mouse looked any different. It really didn't behave that differently. The drug was chosen because of this change in brain function measured by electrophysiology. Right? You get a lot more information about specific pathways that may be altered with this level of analysis than if the mouse twitches. I do a lot of behavior, and we do a lot of behavior. I don't want to dismiss it entirely, but There are plenty of good mouse models for autism where the behavior is really uninteresting, like Fragile X mice. The level of behavioral changes in Fragile X mice is actually quite modest. And people spend a lot of time to try and find like one thing that was different. It was like, you know, they have slightly more accelerated extinction of inhibitory avoidance. True. All right, it's not Fragile X. Fragile X is a pretty serious disorder, and it's not just, you know, you don't have to look for it with a magnifying glass to see it, right? Okay, so SHANK3 is a bona fide autism gene, and it looks like it's probably the most common, maybe second only to FMR1, the most common monogenic cause of autism we know of today. This is 3 families, one with a de novo deletion. So this is where the whole chromosome, the whole part of the chromosome is lost in the boy. Here is a family where they have a point mutation that led to a change in the protein so it no longer functioned. And here is a family where dad has a translocation, and so two chromosomes swap their ends. And so this child has 1 copy, this child has 2 copies, and this child has 3 copies of this region of the genome. We have 2 copies of every gene except on our sex chromosomes if we're boys. But you get 1 copy from mom, 1 copy from dad, right? So monosomy, having only 1 copy of SHANK3 is what causes autism. Disomy is the normal situation. And trisomy, where you have 3 copies, this boy has Asperger's, right? So for a lot of these genes, we're beginning to see that both too little or too much produces a phenotype, and too much tends to be less severe. So you tend to see more autism if you're missing 1 copy, and more Asperger's or other disorders when you see 3 copies. And Mount Sinai has seen now over 30 families with the SHANK3 mutation. These are some of our friends. And Alex Kolebzon and Lata Surya have led this. And just to show you that using gold standard ADI and ADOS, about 80% of these kids meet criteria for a spectrum disorder by ADI and ADOS. 50% for narrowly defined autism. So here's where we can take the mice and make that mutation. So this is part of the mouse genome, and this is part of the SHANK3 gene. And what we've been able to do is we've taken this part of the gene and we've actually taken it and ripped it out of the mouse, out of the genome. So the mouse should have this 4, 5, 6, 7, 8, which is parts of the SHANK3 gene, and the new mouse we created is missing it entirely. Right? So that's the method of gene targeting in mice. And here's the protein, SHANK3, the full-length protein. When you miss 1 copy, you lose about half the protein, and with both copies gone, you have no more protein. Right? So you can actually disrupt any gene almost without exception in the mouse these days. And here's kind of just to convince you, not only did we knock out the gene, but we actually knocked out the part of the gene that we see in 2 out of the 7 families that have known mutations, which is this area over here. This boy has a point mutation in that area, that's him. This child has a point mutation here that actually deletes the same region, right? So, you know, we try to be as true as possible to the clinical genetic findings, because we want to make a mouse that is as close as possible to the human findings that, that's obvious, right? So now here's that electrophysiology I said, which I really trust as kind of a good entree into understanding the effects of a gene mutation. This is measuring the connection between 2 nerves in the brain. Okay, so you stimulate the upstream nerve and you measure the response in the downstream nerve. And what you typically see is that, you know, as you increase the level of the stimulus, you get a stronger response in the post. So, and here you can see this is the wild-type animal, the typical animal, and the heterozygote is missing 1 copy. Again, that's what the human patients have. They miss 1 copy, so we focus primarily on the heterozygote. And what you can see is they look kind of similar. The curve for the wild-type animal is very much like the heterozygous animal. However, this is the same study, but now looking at a different receptor. This is looking at 1 kind of glutamate receptor. Glutamate is what MSG is supposed to give you Chinese restaurant syndrome. Right? But it's a chemical in the brain that signals between nerve cells, right? Nerve cells talk to each other chemically at some point. And so no difference on 1 kind of glutamate receptor. And here in the other kind of glutamate receptor, this is what the wild-type animal looks like. But the heterozygote, you can see it's not, the connection is much weaker. And that tells you something very specific. In fact, this receptor, the AMPA receptor, there are drugs available for it. So it takes us right away to specific molecular targets, right? And we've actually tried the AMPA drugs on these mice, and they actually work beautifully. 2 weeks of an ampakine, and this will go away for the mouse, right? This is another test we did in the mice. This is a measure of cellular learning. So basically, when two nerves talk to each other, they can either strengthen or weaken their connection, right? They can either reinforce a connection, and then they'll be stronger, or they can weaken it, and then there'll be less of a response. So here, the baseline, we chose one of these points over here. Let's say we chose this one. Okay, that's our baseline. We called it 100%. That's the connection between the two cells, the pre and the post. And then we gave a stimulation that's supposed to lead to a long-term reinforcement of that connection. So suddenly, this 100% goes up to 150%. In other words, the connection between those cells is much stronger now. They have a much stronger response in the following cell. This is another way to do it, but let's just focus on this. What you can see is, in the wild-type animal, you have this sustained, for hours, reinforcement. It's a cellular form of memory. It's how we code things in our brains to remember something, perhaps. And the heterozygote, it goes down to baseline. So this important mechanism of reinforcing the connection between nerves is actually not as robust when you lose one copy of SHANK3. This is another way of seeing it. And here you can actually visualize these tiny points are points of contact between nerve cells. We can take the volume of each one with a special microscope, and what you can see is the volume should increase. So when the contact between two brain cells becomes stronger, very typically you see a change in volume. They actually get bigger. That's how it reinforces it. So the volume following this stimulus gets bigger for the wild type, but the heterozygote, it gets bigger, but then it decays to baseline. It's not able to maintain that kind of molecular trace of memory. There are social deficits. So as I said, we do behavior. This is an interaction between adult males and a novel adult female. And what you can see is their social approach and social sniffing, and they also make these ultrasonic vocalizations, so they call to each other. We just don't hear it, and I'm sure you're all glad you don't hear the mice squeaking in your back, but you can get some bat detectors. If you've ever gone out to look for bats, you can buy these, any kind of hobby store, you can go and listen to bats squeaking. So the same things, you can go find your mice. Anyways, those are ultrasonic, so they're outside of our range, but these bat detectors will bring them down for you if you want. And what you can see is the wild-type animals make a lot of calls in a certain period of time, and the heterozygotes make about half as many. The wild-type animals come over and do social sniffing, which is how they kind of get to know each other, and heterozygotes do less. So there are social alterations in these mice. There are also motor abnormalities, which I'll come back to. Oh, I didn't put the slide in. I forgot. Sorry about that. But this is another drug. So I told you that the ampakine drug worked. Okay, ampakines are not, although they exist and many companies have made them, they're not quite ready for prime time. They've not been used. They've not been approved for use for anything. They've been tried in ADHD and a few other things. They're just not there yet. Another drug called IGF-1 is used already in kids with short stature syndrome, a certain kind of short stature syndrome called Laurent dwarfism. That was probably the main reason we tried IGF-1. It's one of three reasons. One is that one, that it's already approved for use in kids. So if it worked in the mice, we would have an easy path towards actually trying it in kids. The second reason was that everything I showed you about that nerve terminal changes suggested that the nerve terminals were a little bit less developed. And this compound was known to help nerves develop a little bit. Okay? So we said maybe it would actually target something we saw as abnormal or different. And the third reason was the same compound, I'll come back to the end, had been tried in mouse models of Rett syndrome, and we really want to find things that work for more than just one kind of autism, right? Okay. So this is 2 weeks injection of IGF-1, and we did a lot of work on this. We started with another compound, which is not a clinical compound. We got great results, and we had to turn around and do it with a clinical compound. And we used, so I'll take you to this slide. So this is what I showed you before. This is, in black is the wild-type animal. You give a stimulus, and you reinforce the connection between the two cells. In white is the heterozygous animal with the SHANK3 deletion going back to baseline very quickly. In red, this is the clinical dose of the compound, it's approved for use in kids today at 240 micrograms per kilogram per day. And what you can see is after 2 weeks injection, the red line and the black line pretty much overlap, right? So we think, changed the development of the brain in that mouse just enough to normalize it so it now is able to make this kind of long-term cellular trace of memory. If you use half the dosage of 120 micrograms per kilogram, so now less than the clinical dose proof in Lorraine dwarfism, you see a tiny effect, but it's not significant. It looks more like the baseline. So we think that we have now kind of evidence not only of that it works, at least in the model system, but also some sense of dosing, right? And fortunately, it's at the same dose that's approved. What I don't have here, and I don't know why I didn't put it in, these mice also show a motor deficit. So they actually are a little bit more clumsy, if you will, which is seen in autism and seen quite a bit in Phelan-McDermid syndrome. The same treatment of 2 weeks shows a reversal of the motor deficits in these mice. So you see, we normalized that electrophysiological deficit I showed you earlier, that curve that was suppressed. I didn't show you this, but that's normal now. This has been normalized, this learning and memory deficit, and the behavioral deficit has also been normalized by 2 weeks treatment. We felt we had enough to go to the FDA. Now, not by accident, we chose this drug, right? Most FDA process is years long and almost impossible to start with kids, right? If you have a drug that you want to try, you're going to start with adults first, right? Autism, we think you want to start with kids. But since this drug was already approved for use in kids, we were able to go to the FDA and say not only did we want to do this, but we also said that we are not sure, but we think we qualify for a waiver. A waiver means we actually don't need the full FDA kind of investigational new drug because it's not an investigational new drug. It's an existing drug which we are repurposing in the same age group for a different purpose. And that turns out to be much easier. And so we got an exemption. What that means is, and it took them less than 30 days. What that means is they said, we agree with you. You are doing everything you're doing falls within the label in the box in terms of who it's the safety and so on. And so we're okay with you just going forward. You'll notice that this is actually, to Alex Koleb's this is actually for a double-blind, placebo-controlled crossover trial in children and adolescents with the 22q13 deletion syndrome or SHANK3 mutation. Okay? As I said before, we want to make an impact on autism broadly. And so at the same time, or once we got the first one, actually we went back right away and said, you know what? we would definitely want to try this in kids without the SHANK3 mutation. We have a lot of data, which I won't show you, that suggests that this pathway is disrupted in autism beyond, you don't have to have a mutation in that gene to have this pathway changed. Go back to the cholesterol example. There's a tiny, tiny number of people who actually have a mutation in the gene HMG-CoA reductase. HMG-CoA reductase is the drug target for statins. I don't have a mutation in the gene, but statins do wonders for my blood pressure. For my cholesterol levels, right? So, so it's quite possible, and it's certainly been the case in other disorders, that a drug that works for somebody with a mutation in a gene will also work more broadly for people without the mutation. So here it is, same long title there. It's a double-blind, placebo-controlled crossover trial in children and adolescents with autism spectrum disorders. So we turned around and said, fair enough, why don't we just roll them out both at the same time? Okay? And the answer, as you probably guess, is it's expensive. So, yesterday we had a webinar. It was organized by the Phelan-McDermid Syndrome Foundation, which is the advocacy group around SHANK3. And we spoke about this because we have now FDA approval for both studies. And we have some pilot money from philanthropy to do our first 10 kids. And I'm not 100% sure. Do we have IRB as of last Friday? Sorry? Yeah. We will. Okay. We basically have IRB. So, and obviously there's a lot of interest among certainly the people with Phelan-McDermid syndrome, but I hope also in the broader community to kind of roll this out pretty aggressively. We don't want to wait the 2 to 3 year normal cycle of NIH funding to at least get something going. We didn't spend all this time trying to short circuit the process and find a good drug that didn't require an IND that, and then have, sit on our keisters for 3 more years. So that we kind of announced that publicly yesterday. We've been talking about it, but as of yesterday, we felt that we could start talking more broadly to the community. It won't shock you to hear that families want to participate, and many are disappointed that it's a small number to begin with. But we are kind of constrained. We put in a large grant to the NIH, which we'll hope to hear over the course of the year to try and fund it further.
Excuse me, so those 10 are the ones who do have the shank deletion?
Our pilot today is with those.
Just the shank?
We would pilot both. We are very actively trying to raise funds. So that's this idea I talked about earlier. This idea we talked about earlier is personalized medicine. And in this kind of extreme example, you really have to tailor like a different drug for individuals with a yellow disorder versus an orange one versus a red one. Okay? There's a new term called individualized medicine, and that's the idea that it's not one drug per person so much as it's groups of people who share similar pathways, and there'll be like 5 optimal drugs, and one will be for this group, and one will be for the other one. So you still individualize it, but it's not personal, right? That's kind of the difference. So this term has fallen out of favor. It's not, you just heard about it really the first time last week, and suddenly it's passé. And the reason I make that point is, I already made this point earlier, that the same compound, this IGF-1, had been studied in a mouse model of Rett syndrome. This is, for example, the knockout mouse, the mouse model of Rett syndrome with T-stands for treatment or without treatment. You see that they live longer. So in Rett syndrome, there's early death. So the knockouts live longer. They're more active. Their locomotor activity, which is normally down in the animal with the mouse model of Rett syndrome, goes back to normal. Their breathing is normalized. Their heart rate, some of the brain chains are normalized. So there was evidence in a mouse model for Rett syndrome that the same drug might work. And in fact, Omar Khwaja has approval and has begun a clinical trial of the same drug in Rett syndrome. And he's been terrific. We've gotten a lot of information back and forth. He came and visited, and we've been in touch with him. And the idea then is we also can learn from each other, and we can also learn about safety, right? So IGF-1, we know everything we need to know about safety in a typically developing child. We also we always are a little bit more concerned in a vulnerable population. So Rett syndrome is not Shank 3, but we know he's done 24 kids already, is that right? And we know the entire list of adverse events, and there's nothing unexpected, right? So we're able to kind of partner with other people and ask questions and learn from each other about safety, ultimately efficacy, and so on. And that's that idea, as I said, of now individualized medicine, that it might be that there are classes of compounds that work better for certain kind of classes of genetic mutation. All right? So I'm pretty much done with everything that's going on. I'll talk a little bit about a new collaboration because I think this gene discovery thing is very, very important. I really do. I'm a believer. And I honestly, in schizophrenia, we look a lot at the environmental component in Alzheimer's too. In autism, we're really focusing on the genetic component because we really think that's where the real findings are going to be. So it's not like I have a bias against other things. The data that I am aware of tells me where to go. I try to be data driven, or at least I say that in public. If we estimate, and that we have maybe found about 15% of the autism genes, how do we get to the next level without ups and downs and all arounds and just like you publish 3 and your sample's too small and then yours is too small. So I had this radical idea that we should share data before we even had the data to share. So that was the formation of what we call the Autism Sequencing Consortium. And I don't know what this is, the Mac to PC. This is Microsoft getting back at me. We already know about 15% of autism genetic risk, and there's very good reason that there will be lots of other genetic risk that could be detected fairly easily with a new kind of technology called high throughput sequencing. And so the idea then was, and I apologize for these weird things, the idea then was that if everybody working in this space got together and brought their expertise and their data, we would be able to think together about what the optimal ways are to find the genes, what the optimal ways to call the genetic variation, how to statistically convince each other there was a real finding there, how to do downstream biological analysis, systems level analysis. This is the concept of what we call strong emergence, that the whole is greater than the sum of its parts, right? That if everybody got together, we'd actually be able to get there quickly. Turns out, and many people don't realize this, gene discovery is not the end game. It's the beginning, right? I always talk about trying to get past get to the post-genomic era in autism, where we have enough of the genetic risk that we can just think about mouse models, other models, and drug and target development. Right? So this is the first meeting. It was right around the corner of the New York Academy of Medicine in April of 2010. Some of you have heard this, but people always point out that Joe's the only guy smiling. Everybody else looks kind of grim because they spent a day getting beaten up. The deal was that nobody had at that point any appreciable data in this new space of high throughput sequencing. And I would say we were asking that people agree to share data they didn't even have yet and weren't quite sure how they're going to get. Okay? But everybody signed up because it's pretty easy to kind of give away something you don't have, right? And we have we have 22 groups that have signed today. We have most major funding organizations represented. And this is probably an underestimate. When you think about the number of individual genomes or exomes that we'll have sequenced by the end of this year, well, by the end of last year we almost reached this. It's about 5,000. We estimate now that by the end of 2012 we'll have 7,000 patients with autism where we'll have sequenced every gene in their genome. Right? And for a variety of reasons, and I've got this nice work going on with the Karolinska Institute. I could sequence this all by myself, and I will find essentially nothing. It's unlikely that with a small sample I will reliably find a new gene. It's even hard with 1,000, right? But with 5,000, we actually this group over here and this group over here have already done several hundred trios and together have found a new gene. And by the virtue of putting all the data together, because once we had this infrastructure, we began to share data more freely, we can actually calculate exactly how many genes we're likely to find with this size sample, or with 10,000 samples, or with 20,000. So we can actually say with a lot of certainty now that if we could do 10,000 samples by within a year or so, we'll probably have found up to an additional 100 autism genes. That's getting to the point where we've gotten enough of the variants that we can begin to really talk about the post-genomic era, right? So we can kind of use this amazing new technology to kind of get us to the next step, as long as we can manage the people. The machines work beautifully. It's getting everybody to kind of buy into the model. The good news is that most of us, have been so frustrated and so disappointed with progress that we're pretty much willing to say, you know what, I know right now that this number's not going to do it, so what's the downside? Why not just get to the next step more quickly by collaborating and sharing? And this consortium has generated a lot of interest, and we've been encouraged. The NIH asked us to come down in September of last year to a summit that they organized that included this consortium, the ASC, plus about another 70. It was open to all kind of stakeholders. We had about 100 and some odd participants. And we put together a white paper which we're going to kind of hopefully find a publisher for. But the idea was to really kind of, get all the funders, all the scientists kind of more or less on board. I mean, we don't all agree perfectly. Heck, we barely agree. But broad strokes, we agree pretty well that this is going to be a way of cutting things down from 5 years to 18 months for phase 1 and 10 years to just another 18 months for phase 2. So, it wouldn't be a bad thing, right? So we've been encouraged actually by the NIH to think about putting in a submission for this summer. And we're hoping that a lot of the kind of foundations will kind of partner with NIH and to make a single large kind of omnibus effort to really get us past this. As much as I like what I'm doing, I'm going to like what I'm doing next more. Right? So that's kind of the end. I do think this is true. We can discuss it. That there are a lot of genes to be found in autism. That finding those genes will change people's lives. Right? I mean, Any drug that works will be a life changer, right? And we're talking about core symptoms. That's what we're going for. We're going for the core symptoms. We're talking about social behaviors, language, cognitive behaviors. That's what we're going for. All right. Repetitive behaviors, sure, happy, but there are some drugs that kind of work for that, right? But it's these core symptoms that really have been intractable, where I think we need to kind of push forward. Lots of genes have been discovered. It's really complicated. Most of us don't think about 100 genes at a time. There are methods of systems biology, and Sinai is a leader in that. So we're filtering all of our genes, not me, but Sinai as an institution and some of the people here, taking all the knowledge we have and trying to understand how these genes interact. So instead of having 100 disparate genes, we have 5 groups of different genes that all work together, because genes don't work by themselves, they work in groups. And so maybe this group will be optimized with one drug target, this one will be best treated with a second drug, this one with a third drug. And that's really the point, that I do think, and I didn't show this data, that when you put all those genes together, we do seem to find specific target pathways that are over and over again changed in autism. And the SHANK3 pathway, which is glutamate transmission, seems to be one of those. That's why we think that maybe a drug that works for a SHANK3 mutation will also work in kids without a SHANK3 mutation. All right, and this is some of the places where we share information. We're not as good as the ASF about this, but we kind of try, and Jess and Pat do a good job of kind of updating, but it's kind of like, it's in addition to all their other jobs. All right, so thank you very much, and I'll stop there.